Yes, hello everyone. I'm Kaitlin Laverty. Can you hear me okay? Okay, great. So yes, today I'll be talking to you, and sorry, I'm in Quade Morris's lab at Memorial Sloan Kettering Cancer Center, and today I'll talk to you about our effort to expand the repertoire of RNA binding protein motifs across eukaryotes. Okay. So RNA binding proteins, or RBPs, are simply defined by their ability to bind to RNA. And I'm sorry, this is a bit redundant with the previous talk, but if there's been any shuffling, hopefully this is helpful. So RBPs are very important for post-transcriptional gene regulation. They are involved in the capping, splicing, and polydenylation of pre-mRNA transcripts, as well as the stabilization, localization, and translation of mature mRNAs. Generally, canonically, RBPs bind to RNA via RNA-binding domains, or RBDs, which generally recognize short, specific RNA nucleotide sequences, like this one, for example. And this is actually RBFOX, which you've already heard about many times today. Often, though, many RBPs have multiple RNA-binding domains, allowing them to achieve greater sequence specificity. And in this talk, I will be referring to the portion of an RBP's amino acid sequence that contains all of the RNA binding domains as the RNA binding region, or RBR. And I'm sorry, this makes it a little bit unclear, but I'm talking about the amino acid sequence that contains all of the RBDs. And as it's been brought up multiple times today, especially by Chris Burge, it's very important to learn the binding specificities of RBPs because it helps us do all of these other functional analyses. So one way we can do this is with RNA-compete, which is an in vitro competitive binding assay. So in an RNA-compete experiment, a tagged RBP of interest is incubated with a pool of designed RNA probes. These probes are then pulled down, eluted, and fluorescently labeled. Then they can be hybridized to a microarray to determine the binding strength of the RBP to each of the individual probe sequences. So after normalization of the fluorescence intensities, we can calculate a z-score for each of these 7-mers based on the fluorescence intensity of the probes in which the 7-mer is found. And this acts as a relative binding affinity. The z-score can be interpreted as a relative binding affinity of the RBP for that 7-mer. And the vector of all of these 7-mer z-scores we refer to as the RNA binding profile. We can also, for visualization and summarization purposes, we can take the top 7-mers and create one of these classic sequence logos. Okay, so RNA-compete is a method that was developed in the Quade Morris lab as well as in the Tim Hughes lab in collaboration with one another way back in 2009. And in 2013, this paper was published with the binding specificities of 205 RBPs measured with RNA-compete. One of the most important takeaways from this paper is we can refer to as the 70% rule, which is that RBPs that share more than 70% amino acid sequence identity with one another across their RNA binding regions tend to have very similar sequence specificities. So this is an example of two RBPs, so human SNRMPA and fruit fly SNF. And this is showing you the correlation between their RNA binding profiles, so the 7-mer z-scores. And you can see that they have a very high Pearson correlation coefficient of 0.95 down in the corner there. And they share 73% amino acid sequence identity across their RNA binding regions. So they took these 205 measured motifs they had, those RNA binding profiles, and they looked at the RNA binding region sequences for predicted RBPs across 288 eukaryotes. And they applied this 70% rule to be able to assign these RNA binding profiles to other RBPs or infer their motifs. And they were able to do this for 8,000 RBPs across the 208 eukaryotes. But if we look at this broken down across kingdoms, we can see that the vast majority of these RBPs were assigned to metazoan proteins, and we have very, very low coverage for protists, plants, and fungi. And even looking at metazoa, you can see that only just under a quarter have an assigned motif, whether measured or inferred. So we still clearly have a lot of work to do in understanding RBP motifs across eukaryotes. So to improve our ability to infer RNA binding specificities across eukaryotes, Matt Weirach, a professor at the Cincinnati Children's Hospital, strategically selected a set of RBPs that, if assayed with RNA-compete, would maximize the number of eukaryotic RBPs for which we could infer a binding specificity, ensuring representation of both model organisms, which are often metazoa, but also these other underrepresented clades. And I just want to say that all of the RNA-compete experiments from the 2013 paper and the ones that I'm talking about that are new here were done by Dr. Debashish Ray, who's a research associate in the Hughes Lab, the Timmy Hughes Lab at the University of Toronto. So altogether now we have 379 RBPs across 33 different species with their RNA binding profiles measured by RNA-compete. And here we have the numbers broken down across the 4 eukaryotic kingdoms. And in the white, you can see the number that were measured previously. And then in the darker color are the RBPs that were newly measured for this study. I also just want to point out that with regards to the types of binding domains, RNA binding domains that these proteins have, the vast majority of them are RRM-containing RBPs, as this is the most common eukaryotic RNA binding domain. And then we also have many KH domain-containing proteins. So if we take all 379 RBPs and we look at the Pearson correlation of their RNA binding profiles, we can create this matrix. But I do want to point out that this is not actually clustered by their Pearson correlations, but rather by the percent sequence identity of their RBRs. And I know this is very big and hard to look at, so I'll show you a smaller subset of this plot. And there are a few things I want to point out to you here. So firstly, we can see that the 70% rule tends to hold, where we have very high similarity of their RNA binding profiles, where we have high amino acid sequence identity between RNA binding regions. We tend to have very similar RNA binding profiles, but this isn't always the case. And you can see that here. These two proteins have 84% amino acid sequence identity, but clearly do not have the same RNA binding specificity. And this is very likely because though overall their amino acid sequences are very similar in their RBRs, the actual peptides that confer binding specificity are not the same. And then conversely, we also see proteins that are very distantly related, which have the same RNA binding specificity, although they have very dissimilar amino acid sequences. And again, here, this could be because they share some common peptide that confers specificity, although overall, they are quite different. So if we could just identify the peptides that determine RNA sequence specificity, we could both avoid incorrect motif assignment using the 70% rule and also assign motifs at greater evolutionary distance, improving our ability to predict eukaryotic motifs. So to do this, Alexander Sassa, who's a former graduate student in Quade Morris's lab, developed joint protein ligand embedding, or JPLE, which maps between a peptide profile and an RNA binding profile using a self-supervised linear autoencoder framework. So the embeddings are computed from a joint vector that contains a peptide profile and an RNA binding profile for a given RBP. And you already know about RNA binding profiles, and a peptide profile is simply a count of each 5 mer peptide, including 1 wildcard character within the RNA binding region. So JPLE is trained with a modification of PCA, but it's different in the sense that from this joint vector, we're only selecting dimensions based off of their ability to capture variation in the RNA binding profile and not in the peptide profile. And theoretically, this should suppress the impact of peptides that have no association with RNA binding on the embedding. So we should only be retaining peptides that actually confer specificity. So JPLE was trained on 355 RNA-compete measured RM and KH domain-containing proteins, because those are the RBPs for which we have a lot of, the RNA binding domains for which we have a lot of RBP data. And it took 122 dimensions or axes to capture 96% of the variance in the RNA binding profile, and that carried with it 44% of the variance in the peptide profiles. And theoretically, again, this should correspond to the RNA contacting peptides that determine RNA specificity. Importantly, JPLE defines a protein encoder along with the joint encoder, so we can estimate embeddings using only a peptide profile and then be able to reconstruct an RNA binding profile, and we call this a protein query. And we can get a measure of our confidence with regards to the reconstructed RNA binding profile using the embedding distance, or EDIST, which is 1 minus the cosine similarity between two RBP embeddings in the latent space, so W. And this here is, this here is also supposed to represent the training proteins and W in the embedding. And we can use the minimum EDIST between an uncharacterized protein that we're querying and the closest training protein as a measure of confidence. So we use a leave one out cross-validation framework to assess the reconstruction accuracy of JPLE's protein queries for all 355 training proteins. And this plot shows the average reconstruction accuracy as a function of increasing e-distance to the closest protein, the closest training protein in the embedding. And just to be clear, the reconstruction similarity is measured as the Pearson correlation coefficient between the actual RNA binding profile and the reconstructed RNA binding profile. And then we also have overlaid here is doing the same thing, but just using amino acid sequence identity. So the same way that we do with the 70% rule, where we have a sequence identity cutoff, and we can at that point just take an RNA binding profile and transfer it to another protein if the amino acid sequence identity is high enough. And if we look here at an amino acid sequence identity cutoff of 70%, which is what we do with the 70% rule, and we go down here, you can see that the cumulative mean reconstruction similarity, or the Pearson correlation, the mean Pearson correlation here is about 0.75. And if we look along here to where this hits for JPLE, and then we go back up, we can see there's a huge increase in recall, and that JPLE is equivalent to being able to use the 70% rule, but to use that as the 40% rule, so to predict binding specificities for proteins with only 40% amino acid sequence identity. Okay. So obviously one of the important things, one of the initial goals, was to improve our knowledge of RBP specificities across eukaryotes. And so we've got our 355 measured RBP motifs. And just to note here, I'm just talking about the RM and KH domain containing proteins, because that's what we used for JPLE. And then at this point, because it's been over a decade since the original paper, we have 690 sequenced eukaryotes to work with. So if we take all of the RM and KH domain putative RBPs from these proteins, and we take their amino acid sequences, and we take our trained JPLE model, we can do protein queries, and we use a very stringent EDIST cutoff. So this is more stringent than what I showed you on the previous plot. And we can reconstruct almost 30,000 RBP motifs across eukaryotes. And if we look at this, so this is broken down across kingdoms in percentages. And you can see, and then here, this is all eukaryotes. And sorry, these colors here, this lighter green shows you RBPs that could be reconstructed using JPLE, but also there was a measured amino acid, or sorry, there was a measured RBP with a similar enough amino acid sequence to just use the 70% rule. But then this darker green color shows you which RBPs could only be reconstructed with JPLE. So you can see there's a huge gain in using JPLE over just using the 70% amino acid sequence identity. And that's particularly true in the non-Metazoan kingdoms. So there, I'm sure you can imagine there are many, many things that we can do with almost 30,000 eukaryotic RBP motifs. And I'll just tell you about one of the things that we did, which was to try to examine the evolution of RNA motifs across eukaryotes. So for this, we identified conserved RNA motif groups, or CRMGs, which are groups of homologous RBPs, so high amino acid sequence similarity, but also a shared motif, so very similar RNA binding profile. And we took 53 species, which covered the evolutionary space within and between eukaryotic clades, and which together contained almost 9,000 RM and KH domain proteins. And we put these into CRMGs, and this resulted in about 2.5 thousand CRMGs, but only 831 of which contained 2+ RBPs. So if these ones that contain multiple RBPs contain RBPs from different species, then we can actually place them on this evolutionary tree. And at the predicted most recent common ancestor based off of which species are actually present in this CRMG. And so that's what this is showing you here. These are the counts of CRMGs that can be attributed to each of these ancestral species. And these are the 53 species. And I'm not going to get into this, but this is showing you the number of CRMGs in the extant species. And I will just point out one other thing here, just sort of as a bit of a sanity check. What this bar represents is species that are unicellular, and you can see that generally unicellular species have fewer extant CRMGs, which makes a lot of sense as, of course, they are not multicellular and they won't often have paralogs that are functioning in different cell types. And this is a much simpler plot to look at, hopefully. So this is just 7 species. They're all metazoa or plant species. And this is showing you the number of CRMGs predicted to be in that species or the ancestral species since the last common ancestor of plants and metazoa. And I'll point out a few things to you here. One of the interesting things that we noticed was that this is the last common ancestor of metazoa, and this is vertebrates here. And there were 2 whole genome duplications along this lineage. And we see also corresponding to that a huge increase in the number of CRMGs. And then there's been a much slower gain of new motifs in vertebrates since then. Conversely, in Arabidopsis thaliana, which is a flowering plant, and C. elegans, which is a nematode, we see over the last 200 or so million years a huge expansion suggesting potentially rapid rewiring of their post-transcriptional regulatory networks relatively recently. And one last thing I want to point out that just might be of interest to some of the people who study RBPs in this audience is if we look back here, we can see that there are 19 CRMGs that are found in both plants that are at the last common ancestor of plants and metazoa. And these include a number of RBPs that you would expect. So RBPs involved in highly conserved processes like splicing. So these splicing factors, and then the 2 poly-A-binding proteins, C0-4, which is involved in deadenylation, CSTF2, cleavage and polyadenylation complex. And then there's also another protein that has a motif that traces back that far, and that's RBM42. And it's an interesting one because it's not something that has a really well-established role in any conserved process. So I just thought I would point that out as an interesting thing that we found. Okay, so I'm sorry I've gone a little bit over time, but in summary, we have a bunch of new RNA compete data. We have this new method JPLE to allow us to build this extra large resource of eukaryotic motifs from which we can do all kinds of things like look at motif evolution and some other things I didn't have time to get to today. And I'd like to thank everyone in the Morris lab, especially Alexander Sassa, who's a former, who's now in Sarah Mostafavi's lab as a postdoc, and Cyrus Tam, who's here as well. And please go check out his poster. This is your last chance after this session, is your last chance to see it. And then also the people in the Tim Hughes lab who are involved in Mount Wyrock and our funding. Thank you.
Thank you. We have time for one short question.
Thank you so much for your talk. Considering the fact that protists actually make up the vast diversity of eukaryotic life, and like plants, animals, and fungi are 3 of more than 100 lineages of protists. I came late, so maybe you've covered this, but obviously the data are skewed towards multicellular life. But if you were to have had more diversity in the protists that are either in the database that you even have access to, or if for future sequencing and efforts going into making the protist data more diverse, do you have any thoughts of what direction that might take the findings, given that there are such divergent and polyploid and all these other things, eukaryotic genomes? Thank you.
Yeah, no, for sure. One thing I will say is that this resource and our labs are basically the only people getting any motifs for any of the plants or protists, or, I mean, not fungi, because people do care about Cerevisiae. But for the most part, like this is now, you might have missed at the beginning, but there was basically no information. And now we've gone to at least something. But I do think that and we have focused mostly on like model organisms or ones that people care about due to, their effect on humans, like disease. And I think, we could focus more on those, and something that we are looking at and doing is some viral REPs as well. So I'm not sure. I'm not an expert on protists, but that could also potentially help.
Yes, some more ciliates that aren't pathogens would be awesome.
Yeah.
Like those have